Hi, everyone. Thanks for coming to hear me speak. And what I'm going to talk today is talk about something that I think really everyone in science needs to be focused on, no matter what we're doing, particularly in neuroscience, and particularly things in the medical sciences, people who receive funding from NIH or other type of healthcare-related funding, which is reproducibility, but also its sister, which is translatability. And as you mentioned, we founded a company, Omniscient, or OAT. And so reproducibility in every aspect of science is really one of the most fundamental features of performing good science, because I don't think we have to state that to this audience. And there's been some serious challenges to this. And we could talk a lot about, we're going to dive into a little bit about how do we handle the challenges? What do I think some of the failures that have happened that have led to, papers like this that are in Nature, high profile, where, multiple very high quality groups doing things the way they thought were best interest got entirely different results with the same exact data set. But also, even deeper than just methodological issues and how we process images, how we even think about how we communicate with each other. And as someone who came to this field from an oblique direction, mainly I wanted to learn how to have better maps to the language system, and then this kind of spiraled into other interests that I came on as I developed in the field. So observations that I had very early on coming into the field that I think are really critical for improving reproducibility and communicability. But ultimately, no matter what we're studying or what we're doing, what we hope is if we're studying the brain, we will learn things that will improve people's lives in some way at some point in the future. And for anybody who's been involved in drug discovery pipelines or any other sort of work, we know that the vast majority of insights that seem very promising in the lab crash at the translation to clinical gap. And there's a lot of reasons for this, but if you look into really where these kind of things falter with regards to neuroscience translation, reproducibility is a big one. So we really have to be certain that what we're doing, at least in principle, is pretty close. So not to say that we haven't reproduced everything, but that ultimately that the core ideas are at least happening enough times that we all can safely say that's probably true, or at least some of this is probably true. And medicine doesn't usually lend itself to absolute certainty, but there's things you could say this is probably true because it's been done by multiple people who found similar results. But more importantly, we have to be able to explain this in a way that people who didn't specialize in doing a PhD or having a significant background in the research can understand it and apply it in clinical practice in a way that's somewhat similar to what we're doing in the lab or in a research setting. And more importantly, and really as you look at how to translate observations from the lab into the real world, the explainability gap for clinicians is much bigger than we really have appreciated, particularly when we start getting into the highly complex things that we're working on. And ultimately, if it can't be brought down to something that does not require attending numerous meetings and or doing extensive training in, it's not going to scale very far. It's not going to really make a big difference because we can't expect everyone to have niche understandings of every single thing that might be useful. And finally, this has to be something that can be studyable, and this means scalable into the real world and able for people to explain what they did, where a person on the other side of the globe who's never met them can read the paper and do it on someone safely. And if you get down to it, those are pretty much the rock solid limitations of translation. You have to be able to do something over and over and over again in a way that a medical education program can adequately teach doctors how to do it. Otherwise, this will never help anybody in reality. And obviously, we can't tackle all of this. Any one of those things could be an entire webinar, and that's not what we're specifically here to talk about. But what we're talking about is how connectomics and specifically certain ways of doing connectomics, certain ways of thinking about connectomics, can actually help improve many of these problems. So the two things I'm going to bring up in this webinar is first the need to have anatomically precise nomenclature. And I'll talk a lot about this, and I'll even go into some things about anatomy, because as a neurosurgeon, details about anatomy are my business. But also we're going to talk about the importance of pipelines and the challenges of having reproducible neuroimaging pipelines. But first, we want to try to win the argument here of, do we, as people doing TMS, need to be anatomically specific about what we're talking about in the brain? Now, as a neurosurgeon, I've worked with many, many neurologists. And the thing that we've noticed as surgeons that neurologists tend to think about, and this is even more true when you get even further away from the practical day-to-day reality, is that neurologists and cognitive doctors often think about surgical procedures as binaries, like a medicine. Either you do it or you don't do it. And the reality is a surgery has thousands of steps that can be modified in all sorts of ways. So there's literally, almost an infinite number of ways to do a surgery with a very complex problem. And so these things don't always lend themselves to, binary randomized trials. And one of the biggest things that from doing a brain surgery that you can moderate that has a pretty significant impact is, where in the brain do you do surgery? And this is very similar with TMS. Where in the brain do you actually stimulate? And so, of course, the question of TMS that's come up for a long time is, given that we know that this is not a pinpoint precise treatment, and we're treating a field and not a little area, does it really matter that we get precise? Now, for me, when I entered the field, the answer was, of course it does, because I've done awake surgery on people, and I know that if you put the electrode at one of these parcellations and you find a speech arrest, if you move about that distance that you see to another parcellation, you don't get a speech arrest. It really is that level of precision and how the brain's really organized. And the Human Connectome Project would certainly argue with this atlas that we really need to think about the brain in specific units. And as I'll show in a bit, many of these areas, the boundaries are fairly firm as what the brain thinks about as a network. But more importantly, it's the fact that if you start looking at things like a gyrus as the description of where you're treating, so even if you're attempting to be anatomically precise with something below the level of the connectome, you can run into a significant problem, most notably that the gyruses don't really exist in the way that you think. So while certain ones, like, for example, the Sylvian fissure, everyone has one. The superior temporal gyrus is usually relatively uninterrupted, but other ones like the middle frontal gyrus are almost always interrupted, and the occipital gyrus is so variable in people that we don't even bother to name the sulci in general. And so part of the reason is the brain isn't organized that way. It's organized more close to the way this looks, and this is what the data tells us, is what Brodmann tells us, and that the cortical sheet really is more of a definition of function than sulcal and gyral boundaries. And so realistically, for us to be able to communicate, what did we test, what did we do, what worked, what did not, and to be able to reproduce these results, we have to be pretty specific of what we were doing, just like a neurosurgeon would have to be to explain what brain approach are we studying when I present these outcomes. So when I got into the field many years ago, and it's particularly after the HCP Atlas came out in Nature in 2016, is we realized this is a really useful tool to do anatomy. So we wrote a lot of papers. This ended up in the neurosurgical literature, where we mapped almost everything you could think of down to the level of really tedious details. And part of it was I just wanted to have a map of what everything talked to everything, where we could go down and use this to build up and do gross anatomy, because ultimately physiology plus anatomy equals medicine. And so ultimately, as a surgeon, I felt that this is something that would be beneficial. And of course, the first question is, does this level of extremely granular anatomy matter? And this is a paper that came out in Brainstem a few months ago actually now. And it was written by Alison Rosen, but you can see there's a lot of people on there. And essentially, these are people who participated in a failed TMS trial. And what they went back, of course, there was image guidance involved in this, so they knew what they had treated. And what we found, if you look on the right side, because we helped her analyze some of the data and look at this in as much detail as possible, that first thing is that even experts were treating pretty significantly different areas in the brain, all thinking that they were treating the DLPFC. And the second is that there was different clustering, and more importantly, that the non-responders were more oftenly treated outside of area 46, and the responders were treated more often in 46. So getting aside the possibility of interperson heterogeneity and all these other things, it really just shows that, quite simply, people were treating different things and that mattered. And if you look at the bottom, if you look at when we look at the average maps, what are the brain networks that get stimulated when you hit these two sites? One is basically in HCP nomenclature, area 8AV, and the other is area 46. They're right next to each other. And the difference is the difference between the non-responders, you're stimulating the default mode network, and then the salience network is what you're stimulating when you hit area 46 in the responders. And so what this argues is even that level of tiny difference matters in what brain network is actually getting activated, and it matters clinically in the fact that this was a failed trial that was a result of even experts treating things without having very anatomically precise targeting. And if you look at these two areas, you can see this is on the HCP map, and even in other studies, and this is a study on the upper left of people just looking and saying what part of the DLPFC are experts treating if they're not using image guidance, and you can see that the scatter is all over the place. But that difference is the difference between hitting two totally different brain networks with two totally different functional and structural connectivity patterns. And so while this is not pinpoint precision, it does suggest that you want to be close, and that basically just being somewhere on the frontal lobe that's 5 centimeters in front of the motor cortex is not all equivalent, and that equivalence in details matters. A further thing to dive into is if we start looking at failed studies and we start looking at responses, for example, in this person who has a stroke, again, without knowing the integrity of the corticospinal tract, which in this case is significantly injured, what makes us think that TMS is going to reconnect that? So it's unlikely. And without having an idea of the structural data and are we even treating an area that's abnormal in this person, it's hard to think that we can actually reduce the complexity without having precision. So what you can see in this particular case is we looked at the structural and functional connectivity in this individual patient, and for a variety of reasons that I don't have time to go into, because this case could actually take up the whole hour if we went into describing why we picked the arrow, which is the contralateral supplementary motor area. We had a pretty significant radiographic and it aligned with a significant clinical result. So it let us have realistic goals that we weren't going to focus on motor, we were focusing on language, and to really get an idea of what the architecture we were working with is. So it isn't surprising that if we think about TMS as a pill, which is you give it or you don't, then we're not actually doing justice to the richness and complexity of the type of treatment we're providing. I think another thing, when we really start thinking about randomized trials, is we're dealing with an extremely heterogeneous machine, which is the human brain. And so this is a picture of my wife on the left with Grammy Award winner Brian McKnight on the right, a charity fundraiser. And if these two people showed up to be in one of our studies, they could be randomized head to head. And of course, we know that their connectomes have differences just for gender alone, but we could go into a lot more details of all the ways they could be different. Even if they were twins, they could be different. And furthermore, we know they have different genomics. So we think we're doing this trial, but in reality, because of the heterogeneity we're dealing with, we're actually doing that trial. And so, of course, when we start thinking about bringing really detailed data into our discussion, we want to really aim to be doing that trial, which is smaller, quicker, and more focused. That's the vision. Now, we know that there's a lot that goes into developing those kind of things, but the computer software started to reach the pipelines where we can actually start to at least know the heterogeneity we're dealing with, begin to control for it, maybe not necessarily limit through randomization, but other strategies. But it also argues that if we do small underpowered studies, we don't have a chance of doing something reproducible, because unless it's something that's so fundamental to the biology of the brain that it's overwhelming and not heterogeneous. So, for example, the motor threshold, people have them, but we all know they have them at different thresholds. And even that is the most consistent anatomy in the human brain. So when we start talking about more higher cognition, we really are underpowered to be able to handle this without more detailed thinking. So one of the things about, if you start thinking about the challenges of doing reproducible science, given how challenging and costly it is to really be able to do enough people to handle heterogeneity. And if we start hanging our idea that everything needs to start off with a randomized sham-controlled study as the first step, we ignore the fact that, machine learning has allowed us to have different approaches to thinking through this problem, where what we might do is try to find patterns first as we treat, develop a hypothesis, and then use trials and random, bigger, more expensive studies that require a lot more effort to validate that, as opposed to starting with that as the first step that we do in TMS research. Because the reality is, if we were to look at every possible combination of every possible target for every possible situation with the heterogeneity we're talking about, none of us will ever be there when anything gets realistically discovered that we have the best target. Not a good target, but the best one. And so when we start talking about reproducible science, the first step is we need to have power. We need to acknowledge the complexity of what we're doing, and we need to be very precise in what we're actually doing so that we can communicate and someone across the world is treating the same basic part of the brain in people and handling the heterogeneity to the extent possible, particularly when we start talking about disease individuals. But even normal individuals, as you can see, trivial differences in position can have a pretty significant difference in what we're doing. But there's also just the simple point of, if we're underpowered, more importantly, if everyone is getting together and stringing together pipelines based on what they happen to think is the best practice and not validating, testing, or interchangeability, maybe not even always doing the best practices, and maybe having people stringing together things that aren't software programmers where things can be done differently than people think, it's hard to believe that we actually, we would ever expect that we would get identical results between centers. So one of the steps that I think is a simple thing, and this is one pipeline that obviously we've spent a lot of time refining. There are obviously other pipelines, but the simple bottom line is that we probably need to be using similar pipelines so that at least we take this out of the situation. There's only a finite number of correct ways to do some of these things. We can argue about specific details, but the idea of having everyone make boutique beer in their own brewery and then expecting them all to taste the same is not realistic. But I think there's a deeper thing, which is if we look at bold signal and we look at the level of connectivity of, if we look at functional connectivity and attempt to compare it in every area, knowing that the bold signal is inherently noisy and there's lots of artifacts that need to be cleaned out, and we're looking at very large data sets when we really start to get down to comparing 379 areas where you compare everything, which is what our pipeline does, and a lot of omics type pipelines are aimed at. We have to acknowledge that it may be hard to recognize that we're not hitting the target precisely, because what's the ground truth? What are we validating it against? And there's an even bigger challenge. And the bigger challenge is recognizing the challenges of making software as opposed to algorithms. So I'll kind of take you into it. For example, we have a medical device arm. We make medical device products under regulatory control. And there's some things you learn about building a quality system for medical grade software that really are eye-opening and aren't something that are intuitive. So when you make software, you first start off by making an algorithm. And then what you do is you engineer that algorithm to run fast, make it interchangeable, lock the code using things like Dockerization, get things to run efficiently and fast. You then build it into a platform these days so you can take advantage of cloud benefits and other efficiencies. You build a UI, UX, make it as usable and understandable as possible. And most importantly, you test every single step down to the unit level and have external validation. Now, why is that the standard? Now, first thing is just, software is extremely complicated, has many lines of code. But there's a bigger thing, and it comes to the concept of emergent phenomenon. So to give you an example of an emergent phenomenon, this is when the sum of the parts yields results that are different than can be predicted from each individual part. And so, for example, a while back, we were baking software that did DTI, and we noticed after looking at dozens and dozens of scans with multiple reviewers that somebody caught that 2 out of 50 scans had a slight 2 millimeter shift that you would only look at if you're really staring at a lot. So someone caught it. And we looked at it. First, we didn't believe it, but it was real, and it was always down in the Z direction. Now, when we went down to troubleshoot this, taking a team of professional, experienced software engineers, this got down to the level of working with how the bits were translating code between Linux and Mac to figure out something at the 17th decimal point. And that round-off error built up around the code to lead to that shift in the affines. So if that doesn't raise the fact that putting things together, you might be getting results that you aren't necessarily aware that they're off, then I think that that's a really good eye-opener. One of the challenges is when we start having everyone stringing things together is even if they're fairly knowledgeable people, besides the fact that emergence can come out of it, we're losing things like efficiency, because what people do is they publish their algorithm, they put it on GitHub, they run a course, they might put a UI, UX if they have time, but there's no engineering and there's not really significant unit level testing to make sure that we're getting something that is what we think it is. Because even if the algorithms work, they might work differently when you put them together in that particular pipeline. So getting past that, even if we are able to do reproducible results and we're all using the same pipeline, as I pointed out earlier, this becomes a significant combinatorial explosion. So we could be doing thousands of trials with millions of patients to answer a single question, like what is the best target for a subtype of depression? And so ultimately, if you think about it, given that the distinction between two areas that are right next to each other can be the difference between this being a depression target and an anxiety target. And I'll show you, for example, that when we were using an agile approach, looking at functional connectivity on an individual basis, we were able to identify that this area 8AV was abnormal in our anxiety patients specifically, and that when we turn that down with CTBS, these are the results to the anxiety scores, which are pretty significant, that even in a small cohort, we now have a hypothesis that this is a good target for anxiety without having to look throughout all the connectome and validate this. And we can start to throw out hypotheses a lot quicker when we start with data as the first step, initiate it on small exploratory cohorts, and get to the answer a lot faster before we invest time and effort in more expensive studies. And so when we looked at this area, why is this an asymmetric target? So why is this distinction that? And it turns out that the target that this is one of the 5 areas of the human cortex that has an asymmetric network affiliation between the two sides. So area 8AV is part of the default mode network on the left, as you see, and part of the central executive network on the right. So you can see on the left side, the DMN areas are in green, and on the right, it's a red area, which is CN. And so the fact that you have this little tiny distinction leading to an entirely different network that's asymmetric between the two sides really should give pause to the idea that the 5 centimeter rule is a good way to even really define the DLPFC, which is a highly complex area. Going deeper into this, when we start looking at treating symptoms, or what is the best strategy for tackling symptoms, the best way to think about, what does a response even mean, is we have to go back and start thinking like clinicians, right? So ultimately, very rarely, if a patient comes in with some combination of these symptoms and schizophrenia, that all of them are equally bad. And that if we, if the number goes down from 10 to 5, that they're half as impaired. Maybe that the one that's giving you the 5 points is the one that we wanted it to go away more than anything, like delusions or something. And so one of the things we have to start getting down to is the level of granularity between symptom and circuit matching, so that we can get past diagnostic labels, so that what we're talking about is this symptom has these specific circuit problems, and these specific circuit problems do or do not serve as the best target. Of course, if we know where the circuit is, we can intervene. TMS being one of many potential options that you could think of of how to modulate a circuit. But again, ultimately, it's possible now to use these kind of large-scale pipelines to turn loose the power of big-scale cloud computing and machine learning to run really interesting machine learning approaches. In this particular case, I set for our data scientists the basic idea that when we made this algorithm that we could not have a black box. So we couldn't have something that didn't tell us what part of the brain to look at. And so, for example, if you look at boosted trees, it gives you a very complex decision algorithm. We don't want it to tell us the person has the symptom, yes or no. That's not a useful prediction. It's where in the brain are you using to make that determination, because these seem like good targets. And so you can see we can crack the black box open. We get a list of edges, which each one of these are 2 different areas that have an abnormal functional connectivity. And we can start to look at a pretty simple question. And this is some data that we worked at with a group at Monash University. And what we were looking at was if they were treating people at the standard DLPFC target, and they actually hit it at area of left 46. They hit the target they expect, which this group uses image guidance, and they did this 95% of the time. So pretty good, given that they didn't have connectomics when they did it, that they were fairly consistent with where they were hitting. But still, even 5% of the time, they're treating a neighboring area. And if you look at that, these are different brain networks if you get down to it. So even those distinctions meant that they were off at 5% of the time and then hitting a different network. It's still pretty good. So we then asked the machine learning to tell us what parts of the brain are most useful to you in determining who is going to have an overall response rate to TMS if you hit area 46. And surprisingly, even though the data set wasn't very big, we had an accuracy of 88% with the machine learning model in replication. And it told us that these 5 areas are what it uses to make that determination. Now, the biggest question is, are these ones that tell you that you shouldn't do TMS? Are these ones that tell you you shouldn't do TMS at 46? Maybe they're the target. Or are these additional targets that need to be added to left 46 treatment to unblock it? This is not known. But what we now have is a clear hypothesis about something that nobody was thinking about, of what is a good other part of the brain to interrogate. Because, again, like what we know with genomics is if you don't know where to look, then you may not necessarily think about exploring that. So again, diving into the anatomy of the connectome, it's worth doing a little bit of detail of this. And what I'm going to try to do in the remaining time I have is to convey some of the knowledge I know about the gross anatomy of the connectome. So there's lots of experts on other aspects of the connectome, the people you can hear talks on that I still am impressed by. What I think I can value add to people's knowledge is really getting into the nitty gritty about what we know, who is part of what network, and how do we really get a visualization of where exactly in the brain this is. And again, as I noted, when I first started getting into this kind of research, the first step I always did try to do is say, well, where should I not cut? And I couldn't find maps of where these areas were that people had lighting up on fMRIs. They would show inflated brain, and again, they have different goals, and they were making it for my purposes. But I do think that as we begin to get more specific about what we're talking about, it's important that people have a basic idea of nomenclature, and we thought this was as good of a nomenclature as any other one, most notably because it uses familiar terms. So V1 is V1. That's familiar to most people who have a neuroscience background. Area 46 is a Brodmann's area. And so in many cases, these have names like P and F and T. These are things that describe lobes of the brain. The interparietal areas have IP in this, etc. But I think there's another couple things that were surprising to me as with it coming from an anatomic background. And if you stare at this map, these are just interesting findings. So for example, if you were to look at 44, which is Brodmann area 44, aka Broca's area on the left side, you notice that the undersurface, and these areas here are sitting on the flip side of the gyrus, the part that you don't see when the Sylvian fissure is closed. They're doing something totally different. I mean, they're sometimes related or part of the same network. I'm sure they communicate with each other, but they're not part of the same network all the time, and they have a different function. And so that level of incredible detail of how the brain divides up its work is fascinating, because in other areas, these areas don't have these tiny distinctions. Furthermore, there are areas that actually cross gyruses. There's areas that sit inside side, like the callosal sulcus, that we don't pay much attention to. And ultimately, this is a real statement by the brain that it does not care what we call a gyrus. This is not a way to describe brain anatomy. Even though it's useful, it's simple, and it's easy to learn, and probably we should continue teaching it for just simplicity's sake. We need to really start getting past the idea that a gyrus is the unit of human function in the brain. It's just not. Now, this is work by Randy Buckner and Tom Yeo. Probably some people have seen this. I think that it's a really helpful starting point into getting at the anatomy of brain networks, in part because they basically define that every part of the brain could be roughly affiliated with about 7 networks. That was the solution they used for the machine learning tools to divide this up. Some of these are fairly simple unimodal, like sensory motor, vision, or areas like limbic that are fairly familiar to people, with the default mode network, the central executive network, salience, and DAN. Usually to clinicians, these are completely new, or they've only heard about them briefly. Now, how they define this, for those of you who are not familiar with these papers of how networks were defined, is generally, if we look at different bold signals in different locations, they're never going to be perfectly overlaid. However, what you're going to often find is that many of them are very close to each other, and some of them are very opposite each other, meaning one's up, the other one's down. And by forcing the tool to define the boundaries of different regions based on similarity, you can basically divide the brain along different axes. But it turns out that if you set a more stringent threshold, there are other solutions that require the brain can be continued to subdivide. So in the Buckner-Yeo system, this was 17 areas or 17 networks, and this really sees the default mode network, the vision system, and the central executive network splitting. But also this can go down all the way to the level of parcellations, where all the parcellations are more similar to themselves than anyone else. I think it's a helpful tool for people who are not deep into this science to kind of make it approachable. And if you look at what happens is ultimately there's a core portion of the network. So the default mode network, what everyone thinks of as the canonical, which is the anterior cingulate, posterior cingulate, and lateral parietal, could best be thought of as a core with some other areas breaking off. And often I found that when I first encountered this was actually very confusing to me, because some people's DMNs looked a lot bigger than others. And oftentimes it's not having precise terms and nomenclatures for how we describe these things, where did this term come from, and what does this really mean? So what this means, if you look at, for example, on the 17 network model, there's the red areas at Broca's area, the temporal lobe, and a few other areas that turn out to also be language areas. The language system is part of the global DMN. It's closer to the DMN than it is to the central executive network, but it's closer to other language areas than it is to non-language areas. And again, this is reducing a high level of complexity to a single slide, but I think it's a helpful way to think about it. But when we start getting down to the default mode network, if you really start to look at this at the parcellation by parcellation level, it's a lot more complex. So the DMN is actually probably multiple subcomponents, many of which they do different things. And we know that there's at least 2 to 3 different ways you can split the core DMN into different parts, and these do different things. And even if you having looked at lots of different subsets of a giant connectomic matrix, it's really rare for all of this to be in sync with itself and all anti-correlated with the central executive network. So while these things are very easy to do, to look at whole network models, we probably need to move more granular in some cases to find some of the features we're looking for. If you look at this, the language system, again, this is the first reason that I got interested in this field, because doing awake brain surgery, I found that even having a very good training how to do these operations awake and what I thought was a good understanding of the language system of the human brain, that patients woke up with language and speech problems that I didn't totally understand, and ultimately the model was incomplete. Most notably, the Human Connectome Project identified this area 55B. It's in the middle frontal gyrus and not a place that we think of as canonical Broca's area. And we've actually looked at a few cases, and several other people have repeated this by looking at their cases. And there are people who develop apraxia of speech when they have injury to this. There's also a different cell that suggests this is a motor cortex of the larynx. And ultimately, this is obviously a speech production site. And it isn't something that we've been thinking about as potentially a target, but anecdotally, I've done this in a few people who had functional anomalies, and this can be very helpful. So by looking at a more detailed anatomic system and being specific, we can communicate that, hey, maybe this is something we should think about as a target, and we can all go back and do the same thing if we're utilizing connectomic tools to actually pick our targets. Now, if you start looking at the other portions of the default mode network, notice that the global auditory pattern, the higher areas being A4 and A5, the non-primary areas, they actually affiliate differently, but the more lateral SCG areas and parts of the posterior parahippocampal gyrus do different things within the global DMN in addition to language in the core. And so again, they're more close to each other than they are to the limbic system, but they're more close to their own subcomponents than they are to the other parts of the DMN. The salience network is a really interesting, important network. It's one that we as neurosurgeons didn't know about because we weren't attending the same meetings. This information didn't get communicated. What we learned from this very quickly is if you injure this connection and it's a junction with the default mode network in the middle cingulate region, you leave people akinetic mutes. This has actually also been independently suggested by Michael Fox's group, who showed that there is this little area around the junction of the bend of the anterior cingulate gyrus that seems to be very important for initiating movement. And this was a really significant step forward in neurosurgery when we learned this, but also that notice that it has this connection to the anterior insula via the frontal ascending tract. We've also recently published a paper demonstrating that cutting the frontal ascending tract, or more importantly, trying not to cut the frontal ascending tract when doing surgery, eliminated the supplementary motor area syndrome in our practice. So this is probably also part of an initiation system. And finally, when we look at the central executive network, this is a simplification. This is only the core section of the frontal polar region, this area PFM and the supramarginal gyrus, and area 8C. 8C is a hub of the central executive network and joins them up through this kind of long SLF connection. Now, again, this is kind of rushing for people who haven't dived into this, and we can go as deep as people are interested in over time, but ultimately this is, I think, a good overview of where this actually is physically located. And again, areas that probably we haven't thought of, like nobody's talking about 8C, but it's a hub of the brain. No matter how you slice it, it has one of the highest centralities in the cortex, and probably this is due to its role linking up portions of the central executive network. The dorsal attention network is a network that I think that is, the more I learn about it, the more I think that we probably needs a new name. It was discovered in attention, maintaining directed attention, but of course, it probably does a lot more than this. And of course, the challenges that from a clinical standpoint, we know that I can just tell you anecdotally, if people who had tumors that grow into that particular connection in this general area, they definitely look very differently. Their cognition drops significantly. But more important, it's hard to disentangle this from all the other areas around there. And I think it needs further study to figure out what is the clinical significance of injury to the dorsal attention network. In addition, when we think about the sensory motor system, we think about the motor cortex. But of course, we know, we've learned the hard way in neurosurgery that ultimately cutting across interconnections between motor planning areas often has an even worse effect than just cutting part of the motor cortex out. And so as well, of course, if you disconnect the corticospinal tract, or a stroke or other lesion destroys it, the chance that TMS is going to connect that back up is relatively low. And so when we look at studies that have mixed results, we can look at a lot of reasons, but one of the simplest ones is we should probably have an inclusion and exclusion criteria for being in a stroke study with TMS, that we actually confirm that the corticospinal tract is anatomically at least there, because obviously that changes the prognosis pretty significantly, and this is a source of heterogeneity. The visual system, as again, you see that you can demonstrate their multiple components. These are all in clinical grade scans that we perform. Now, the challenge is that, of course, this is so localized and highly interconnected, it's hard to really dissociate from a clinical standpoint. what the right targets are. And of course, what's interesting is that these, looking at some of the large-scale data blasts that we've had, looking at mental illnesses of various kinds like schizophrenia, often when we look at things of difficulty of abstract thinking and things like this, the higher level visual areas, most notably in the WARE pathway, the dorsal stream, tend to show up more than you'd expect. I think the vision plays a lot bigger role in cognition than we probably appreciated. Also, when we start talking about, and there's reports in the literature about TMS as a treatment for neglect, but of course, one of the things that's really interesting about it is, well, where's the target? And for years, people in neurosurgery have just kind of been afraid of the right parietal lobe because there wasn't really a clear definition of what we were trying not to do. We think that we know for pretty certain after doing awake surgery where we've mapped the superior longitudinal fasciculus and the arcuate bend of the supramarginal gyrus, that seems to be necessary to avoid neglect, because when we stimulate it, people start neglecting. When we stop stimulating, that resolves. And furthermore, when we don't cut it, we don't get neglect. And so given the ventral tension network is running through that seems like a reasonable candidate for this. But again, intraoperative studies do have some challenges of being precise with this. And we need to have converging evidence that really starts to point to what these networks are likely doing. Finally, for those of you who are not familiar with the multiple domain network, it's not a pure network by itself. It's actually different components of different networks. Like you can see, there's parts of the central executive and other dorsal tension and other networks. But it's really been it's an interesting network, and there's some data that demonstrate that after a stroke, the cognition drops relatively proportional to how much of the multiple domain network you lose. So I covered a lot of ground. I tend to do that in my talks. And I hope this was informative. I covered everything from reproducibility issues in science to gross anatomy. But I hope what I really conveyed to everyone is the real importance of us talking in terms of anatomy, and more importantly, cross-communicating across disciplines, because often different ways we have of thinking can really cross-pollinate and bring new ideas on ideally what the best strategies we are for therapeutics, and also how do we sync neuroscience and clinical care in a way that leads down the translational pipeline correctly. So thanks, everyone.
Brilliant. Okay, thanks for that, Mike. So yeah, attendees, you'll have got a grasp then on the kind of things that we can start to explore with connectomics. So on that, BrainBox, we know then also that connectomics is a very, it's an imaging heavy resource. Imaging as well is something which is, particularly in the UK, it's a bit of a barrier to having a lot of participants given the cost of it. So on that, BrainBox and O8T, Omniscient, have partnered together to launch, bear with me when my screen starts sharing, the Infinitome Imaging Award. So what we plan to do is offer, similar to some of our other challenges where, you know, the research challenge, for example, where someone submits an abstract and then, you know, is the winner of some equipment provided by BrainBox. The collaboration with BrainBox and Omniscient is that we ask for a study protocol, and that then allows you, the winner of that prize then is granted the equivalent of $20,000 to put towards scanning time, with access to Infinitome software as well. So there's much more information on this on the BrainBox initiative website. So if you head to brainbox-initiative.com/awards, there's a bit of a brief as to, you know, the intended, you know, what the intended study protocol is. but yeah, we will let this run until September the 1st. Will be the deadline for submissions, and then a couple of weeks after that, we'll do a bit of a follow-up chat with probably with Mike and the guys from Omniscient, and then we'll also announce the winner there. So that's a couple of months to pull together a protocol, and then yeah, we'll announce the winner of that prize. But yeah, it's essentially a $20,000 US, or the equivalent in whatever your local currency is. It's essentially an imaging grant. So it's a prize that packs a bit of a punch. So we'll go next on to my colleague Dan's favorite bit of the webinar, and kind of give you a brief as to what else we can do as BrainBox Initiative. So BrainBox Initiative is kind of one arm of what BrainBox as a body can do. And every year we run our annual conference. Obviously, the last 2 years now, we run this online. September will be the same instance, but a number of speakers focus more at early mid-career researchers. There's a whole host of speakers. There's poster challenges, poster presentations, research challenge, and Young Investigator Awards. All get announced kind of in this conference. We'll be carrying on with our webinar series, and the next is on the 28th of July, and it's about the best toolbox. So again, head to the BrainBox Initiative website to find out some more information on that. We have our workshops coming up. So the TES workshop, again, this is all online, with spaces still available for both of these. These are coming up in the next couple of weeks. So not only do we kind of show you, how to use these devices. It's also a bit of kind of one-on-one time with researchers and experts in each of their fields, to help you kind of set up your own studies and any problems, hiccups that you might come across during the process of doing these studies, with also the technical expertise from the BrainBox side of stuff with regards to the products. So the TES next week, and then the following week will be the TNS workshop. As I've mentioned, the conference will be running in September. You can register your interest at brainboxinitiative.com, brainbox-initiative.com/conference. It's £30 this year, so it's a drastically reduced cost, given that it's an online-only conference across 3 days. These are some of the speakers who we'll be looking forward to seeing. And obviously, we have our posters, which will be presented during that conference. Each poster will have a 3-minute opportunity to run us through kind of the research behind the poster. And within the conference, then we'll be announcing the winner of these 3 prizes as well. Again, if you head to brainbox-initiative.com/awards, you'll see these 3. And of course, the newest award, the Infinitome Imaging Award, is on the same page there as well. None of this would be possible, of course, by the other arm of the company we have, which is BrainBox Limited, that hosts kind of all of the products, Infinitome being one of those. So for any more information on any of this information, be it hardware or software, or how to do a particularly difficult technique, so TMS EEG, kind of how-to guides and stuff like that, all hosted on the website. And I think that wraps up there. I'm going to double check really quickly the Q&A. I did look during, and it was just 5 questions saying, Mark, your mic is muted. No more questions. But if there are any that pop up, by all means, please do get in touch. So you can reach me, Mark, at brainbox-neuro.com. There's one just pop up there. Is it possible to ask specific questions? Of course it is. He's still here. Yes, by all means. If you have questions, please do. And I should have said this at the start. I do apologise. If there are questions, please do pop them in that Q&A box. And yeah, we can chat to Mike and put any questions to him. On getting hold of the rest of the Brainbox team, then, so of course I'm available. Mark at brainbox-neuro.com, and the rest of the support team would be support at brainbox-neuro. So I'll give us a couple more minutes to let any kind of questions roll in that we can put to Mike. Mike, is there anything else you want to kind of to wrap up on at the end there?
No, I'm just interested if anyone has questions. I'm happy to answer them.
Cool. I'd love to hear what your thoughts are on the main issues or challenges with converting more conceptual research to clinical practice.
Oh, I mean, I could talk about that for hours if she wants, but the biggest challenge ultimately, if you talk about going from concept, if you really wanted, there's actually a great paper in Nature this week on this, and it was an opinion piece about delivery science. They said we're really good at data science, we haven't got good at delivery science, and it's kind of just something I've been saying for a long time. So, I mean, here's a good example. So let's say you're talking about what is the best imaging protocol to do. Well, in a research scanner, you can do some 120 direction, extremely, all these things, field maps, because it's a different discussion. But if you try to do that on a scalable basis in the hospital, we've calculated out if you just add a resting state of 8 minutes or more, and you don't keep that time neutral, the cost to a hospital that's doing a lot of scans is over $1 million a year. So they're not going to let you do that. So what you have to do is say, okay, let's scale down from the best possible thing to get a Nature paper versus what's scalable in the real world. And then let's optimize noise correction as best we possibly can with this and get as much data as we can and then know where the limits are. Furthermore, the second challenge is ultimately that there's always, when you look at hospitals, people are really busy and everything works on habits. We do this, it works, it's protocolized, standardized. And if what you're doing requires drastic changes to that, it better be something that is so overwhelmingly beneficial that it just would be unethical to not provide it. And what that means is most things are just not that, right? I mean, it's very rare that you have a COVID vaccine, or some drug that eliminates, Hep C, where it's really just, we have to deal with this. This is clearly mandatory. Everything else, what happens is it's a good thing to do, but if it's really too difficult, people are going to not do it. And so figuring about how do you solve problems with acquisition and delivery in a scalable fashion, because if something's really going to, a good example is if you look at TMS. Ultimately, it's drastically underused in clinical practice, given the risk benefit. So as a neurosurgeon, I drill around the carotid artery there's no randomized trial that says it's a good idea to do that. People have done it. We said if there's tumor going into the optic nerve, you have to do this kind of thing. But that's the level of risk that we have to take for some of these people. So comparatively, TMS is so much safer than this. But what hasn't happened is, that information didn't make it over to clinicians. It's made it in the form of psychiatry. We have certain rules about this. And what ends up happening is there's not really a way to think, how do we do this in the hospital for people who are in a coma, which is actually the people who the risk benefit is incredibly clear, like you can't make things worse. And so ultimately, it's the difference between, if we wait for ourselves to have absolute certainty of the brain, what we're going to do is we're never going to put the tools in people's hands to say, well, they're pretty good, and let the clinicians give us feedback and we'll keep making it better, which is kind of our angle. Is why I ended up, I still have an academic footprint, I still publish, as you pointed out, but these kind of problems are things that industry is very good at solving. And what we really need to think about is, how do you bridge and get the people who understand the science to understand what the constraints are, which is not the absolute best possible way that we could do it, but what's a pretty good way that's 99% or 98% of the way there, gets very good results, but is scalable. That's really one of the biggest challenges. EEG or MRI, the answer is, Yeah, yeah. I mean, this is the classic thing. Neither one of the. It's like Spider-Man and Venom. They can't eliminate each other, and so ultimately, if you think about it, EEG has some huge advantages with temporal resolution that Bold cannot do. There's certain things you can do with EEG that you can't do with MRI. And of course, there's the benefit of being cheaper, but the challenges, of course, when you get down to the spatial localization, and when you're talking about clinical practice, and if we're talking about that moving a centimeter actually changes the results and changes what you're actually doing, then we have to ask, is EEG ever going to get that kind of spatial resolution? And so what I've always thought about is if we really had a perfect paradigm that was in the TMS world, and this is my gut, the MRI is going to be where do we do it, and the EEG is going to be aimed at which signal should we be delivering, because we assume theta burst is a really good one and we should do it, but it may not be the best of all possible stimulation paradigms, and it may not be the best in that patient, it may not be the best of the target, et cetera, et cetera, et cetera. We've just kind of written this off because what worked for someone else, so I'm going to keep doing it. And these are things that we don't necessarily want to modify clinical practice, but if you think about something that's a fairly simple low-hanging fruit, we use them for what they're good at. Let's not try to make one thing solve all the problems. And the most important thing is that as much as possible, we should get the source localization as close as we can. So we've actually built some tools that will, if you tell us what the cap is, it will tell us what parcellations you're reading from, usually reading from 2 or 3. So you're at least closer to that target. And ultimately, I think we really have to have a discussion about, anatomy and wiring is the fundamental basis of how the brain works. And we can't ignore the wiring diagram when we think about what we're doing to the brain.
Cool. So, Deepa, Cecile, I hope that answered those questions. There haven't been any more ruled in the interim. So I think we might look to wrap this up. If you ever have a spare hour or so, there's a lot of previous webinar information is also hosted on the BrainBox-Initiative site. So by all means, over a particularly slow lunch hour, you could go and have a look at those. So I guess the last thing to say is, Mike, thank you very much for your time. We appreciate you're a particularly busy man. And yeah, we look forward to reading some of these prize submissions very soon. Thank you all.
Thanks, everyone.